Okay, so this video is looking at the respirometer, which is a piece of equipment which allows us to measure the respiration rate. If I just kind of talk you through this diagram, what you can see is here, what you have is you have got some respiring organisms in here. So that will usually be germinating seeds or little insects like woodlice or things like that. Those aren't insects, but anyway, little small creatures. And they will be respiring, so they'll be taking in oxygen, they'll be releasing carbon dioxide. Now down here, you've got a chemical which absorbs carbon dioxide. What that does is it means that oxygen is going into these little creatures, but the carbon dioxide which is going out is absorbed by this. So that reduces the amount of gas in this tube, which causes a pressure which pulls up this way. And so this fluid in this U-tube will move, and you'll see that there's a scale here, and that then allows us to measure the distance that goes. This syringe will then allow you to reset that tube and repeat the experiment. So going into a little bit more detail, tube A is what we call a control tube. Now, it's important in scientific experiments to have controls, and the control tube is important because it shows that any movement is to do with respiration and not anything else. And so you can see in this example, what they've done is they've increased the volume of the potassium hydroxide. Basically, what you need to make sure is that the volume of free air in this tube is the same as the volume of free air in this tube. Okay, so sometimes you can do that by increasing the volume of the potassium hydroxide, or what you could do, and what's quite common to do, is instead you add another little basket in here, and you just fill it with glass beads that have the same volume as the volume of your respiring organisms. Okay, so the really critical thing here is the role of the potassium hydroxide or sodium hydroxide or soda lime, and that absorbs the carbon dioxide. And you can see in this instance it's a liquid form, and that there's a filter paper that's rolled up to form a wick, and what that does is increases the surface area of the absorbing chemical, thus making the absorption of carbon dioxide more efficient. In tube B, that's the experimental tube. So as I explained, you've got the respiring organisms. As they take in oxygen, you're going to have a reduction in the gas volume of tube B. Now, you would expect that to be matched by the expiration of carbon dioxide, but because this is absorbing the carbon dioxide, you get the reduction in the volume here, which causes the pressure to suck, essentially, this tube and move the coloured fluid. When doing an experiment using this sort of thing, then it is really important to consider a few factors. So you must consider acclimatization. So basically, you set up your equipment, and if you're using living things, you need to give them a period of time to adjust to the new conditions. So they need to have, 10 to 15 minutes to get used to being in the equipment. You then need to consider, do you really need to use animals? So when we look at this, we tend to think, is there a viable alternative to using an animal? Can you use the animal without harming it? Can the animal be returned to its natural habitat? And you have to sort of make a call as to whether using animals is appropriate. In this case, probably not, because you could quite easily use respiring seeds or peas. And then if you do use animals, you need to keep them safe. Potassium hydroxide will kill animals, so you need to make sure that there's a very clear mesh in between the animal and the potassium hydroxide, so that they don't accidentally drop into the potassium hydroxide and drown, and it would be a very unpleasant death for them.